Hello, and thank you for joining us. I'm Kristen McPike, Program and Alliance Manager at the Antibody Society. Today's webinar is one in a series designed to inform and educate our members in the broader scientific community about topics related to antibody discovery and development. Our expert speaker and 2024 James S. Huston Award winner, Dr. Jenna Guthmiller, will be discussing harnessing the memory B cells of influenza past to combat seasonal and pandemic threat influenza viruses today. Please note the webinar is being recorded. Please add any and all questions to the Q&A box in the viewer, and the questions will be answered at the end of the webinar. Without further ado, I'll turn it over to our speaker. Thank you.
Great, thank you, Kristen, and hopefully you can see my slides. I slightly changed the title to sort of be a little bit of a play on a classic book, but more so a movie that I was obsessed with as a child. So today I'm going to talk to you guys about influenza viruses, which are a member of the Orthomyxoviridae family. They are a negative sense single-stranded RNA virus. They possess sort of this unique genome in that it's segmented. So essentially every gene of the virus is expressed on its own genetic segment. And due to the fact that it's an RNA virus, there are essentially no proofreading mechanisms. And sort of through a combination of these lack of proofreading mechanisms, this is a rapidly evolving virus. And there's really two ways that influenza viruses can evolve. One way is through this lack of proofreading mechanisms, known as antigenic drift, in which the virus will specifically evolve to evade host humoral immunity, so evading antibody responses. In contrast, influenza viruses can undergo this process called antigenic shift. This actually occurs when two influenza viruses infect the same cell and then exchange that segmented genome. So essentially you can get a new virus out of the cell that is expressing new genetic segments that have sort of been switched around, resulting in something that is antigenically novel to the population. And oftentimes these antigenic shifts can lead to influenza pandemics. We in my lab are really focused on the two surface glycoproteins of influenza, the hemagglutinin or HA protein, which is essential for entry. And currently, and I keep forgetting to update this slide, there are 19 subtypes for influenza A viruses corresponding to the HA protein. The other protein we're interested in is neuraminidase, or NA. And this protein really functions on the back end of infection by allowing for new virions to be released from an infected cell. Similarly, neuraminidase undergoes the same process of antigenic drift as the HA protein, and there are 11 different subtypes. And I also just want to note, and this picture right here is just highlighting the sort of antigenic diversity of the HA protein, and that there's sort of two different types of the HA protein, or two different groups, I should say. But also, despite all these different subtypes, within every single subtype, there are numerous clades circulating, some in humans, some in other animals, birds, pigs, things of that nature. So there's a huge diversity of influenza A viruses. And as a result of sort of these processes of antigenic drift and antigenic shift, this led to 4 pandemics in about a 100-year time span, with maybe the most infamous of these being the 1918 pandemic, but certainly the most in recent history was the 2009 H1N1 pandemic. Okay, so my lab, we're really interested in understanding sort of how we can generate more broadly protective humoral immunity. And there are a few key barriers when it comes to generating broader humoral immunity against.
Jenna, I'm going to stop you. We are actually having an issue on the end user side, so I'm going to upload your presentation as the handout here. So I am going to stop your screen share, but actually move us to the slide deck. So give me just a moment. Apologize for that. So Jenna, if you click back on where it says slide control if you come out of your screen share, go up to the control panel, hit slide control, you should see your deck loaded there in the thumbnails.
Okay.
And then go ahead, click on thumbnail number 3 for me where you were. Yep, exactly.
Yeah. Okay. So I don't need to start over.
You do not. Nope.
That's great. Okay. So as I was mentioning, that there are several key barriers when it comes to generating broadly protective immunity against influenza viruses. And one of these is this idea of immunodominance. And essentially what that means is that we do not target all parts of a virus equally. And in fact, in the context of influenza, for whatever reason, we preferentially make antibody responses against the variable parts of the HA protein in this case. I'm showing below the structure of the HA, which is comprised of two domains. The head domain, which corresponds to the receptor binding portion of the HA protein, and then the stalk domain, which is much more well conserved. So again, more responses against the head domain, which is more variable, undergoes that process of antigenic drift more rapidly, and less so generating responses against the stalk domain. And then I'll also sort of show some data today supporting that for whatever reason, even when we do try to make antibody responses against conserved portions of the virus, we tend to target non-protective epitopes, oftentimes overprotective epitopes. Another challenge that we face in the influenza field is that every single adult has seen influenza viruses numerous times. It's projected by the age of 20 that you've seen influenza at least 5 times in your life. And really what that means is that past exposures influence future responses to influenza viruses. And this is a term that was originally coined as original antigenic sin. But because that sounds so negative, we've tried to move this as a field to this term imprinting. And what that really means is that you are reusing old memory B cells to fight new viruses. And these new viruses may have mutated to sort of get away from these memory B cells. And as a result, even though the memory B cell may still bind and recognize that virus, they may be of lower affinity and less potent, or lower potency. But this pathway also allows us to generate things like broadly neutralizing antibodies, albeit these are quite rare. And then on the right-hand side, just to illustrate that, there's sort of been a push to generate better, more effective influenza vaccines. And this really needs to go to this approach of, instead of generating strain-specific immunity, which is meant to target circulating strains and is sort of the goal of current seasonal influenza virus vaccines, we really need to be making broader immune responses that can target all influenza viruses within a given subtype, across potentially multiple subtypes, and then potentially across both those group 1 and group 2 influenza A viruses. And because the known correlative protection against influenza viruses are antibodies against that HA protein, maybe the answer to this problem is generating broadly protective antibodies. Okay, so how do we study B cells? And unfortunately, I don't have my animation, so I tried to make this in the context of A Christmas Carol. I'm a big fan of the Muppets version of this, so we'll kind of use the ghosts of influenza, past, present, and yet to come as sort of a theme of, you know, what memory B cells can be used when. But how do we study memory B cells? And so there's a few different ways that we can do this. We can either get samples from people prior to exposure, and sort of fish out memory B cells to understand what does their memory B cell repertoire even look like prior to an exposure. Similarly, we can recruit people that have been vaccinated or infected. And one sort of nice thing is that when we are infected or vaccinated, that initial wave of antibody secreting cells known as plasmablasts are almost universally derived from memory B cells. And so this plasmablast response allows us to dissect, you know, how we're responding to this infection or vaccination and how we're reusing memory B cells. And then the last thing we can do is that we can isolate memory B cells following infection or vaccination to understand how those exposures have sort of reshaped the memory B cell pool. Okay. So just to start off on the ghost of influenza past. So one question that we had, this is actually some work during my postdoc, was to understand, well, how does different types of exposure, so infection versus vaccination, influence what memory B cells are recalled? And this was a project that was undertaken with a graduate student in my postdoc lab, Haley Dugan, where she was working on infection, I was working on vaccination, and we really wanted to address this question of how these things are different, or how the memory B cells recalled are different. And what we found is that when we get vaccinated, we are preferentially recalling memory B cells against that HA protein. And this is representative of the plasmablasts and as monoclonal antibodies. In contrast, when we are infected either by H1N1 or H3N2 infection, you'll notice that the majority of the response is not against HA, and in fact, a little bit more dispersed. You see plenty of responses against that neuraminidase protein, so the other surface glycoprotein that you don't see from vaccination, But you also see a pretty good response against the nucleoprotein, or NP, which is a protein that encapsulates the viral genome. So, very big differences. So we also wanted to understand, well, these ideas of imprinting or original antigenic sin, how is that influenced by whether you're infected or vaccinated? And essentially what we found is we just tested these monoclonal antibodies for affinity against any past strain, relative to the current strain that they would have been infected with or that they would have been vaccinated with. And what we found was that with the infection, that they were much more likely to have stronger affinity to those past influenza strains than those B cells that were called by vaccination, really suggesting that you're reusing really old memory B cells. And furthermore, we did test sort of what we thought was their imprinting influenza virus, so the very first influenza virus that they were exposed to, and this still held up in that infection-induced antibody responses were more likely to bind a childhood strain than those induced by vaccination. And what that really suggested to us is that perhaps infection is recalling basically any memory B cell you have, whereas vaccination is potentially recalling more recently adapted memory B cells. In addition, when you look at neutralizing versus non-neutralizing, not too terribly surprising, the vaccination-induced responses are much more likely to be neutralizing than infection-induced. And then if you look at the potency of those neutralizing antibodies, again, vaccine-induced responses are of higher potency of neutralization compared to infection. So what this really highlighted to us is that infection really follows these rules more of the original antigenic sin, so this negative aspect, where you're reusing old memory B cells, in this case we think really old memory B cells, to combat new viruses, whereas vaccination perhaps sort of is using more updated responses in order to provide that protection. Okay, so then what about in the context of influenza? How does pre-existing immunity shape how we respond to different influenza vaccines? And really, I was very fortunate as a postdoc to have access to samples where we had individuals that had low pre-existing immunity to a virus prior to vaccination. So this was in the case of people that were receiving the 2009 monovalent influenza vaccine. So this was the vaccine against the pandemic 2009 H1N1 virus. And then we had another cohort of individuals that we sort of predict had high responses. And because I don't have animations, I guess I won't be able to show you the data that's going to be underneath the low or the high. But just trust me when I say that when you have low pre-existing immunity against, in this case, H1N1, you preferentially recall memory B cells against broadly neutralizing epitopes. In this case, we were specifically looking at these broadly neutralizing epitopes of the head domain, the receptor binding site, which I've outlined sort of in pink on the left-hand side, which is that sort of tiny little conserved portion, and then the lateral patch, which is that red, sort of larger red portion. And again, you'll have to trust me when I tell you this. On the right-hand side, I'm just presenting this as the number or percentage of RBS or lateral patch targeting antibodies per individual that we studied. And again, really strikingly, if you had low pre-existing immunity, more likely to recall memory B cells against these conserved epitopes, whereas if you had high pre-existing immunity, you didn't. But you also notice I have sort of that person that's the red square around. And this was a person from the 2014 cohort. So these were individuals 5 years after the pandemic. It's projected about 90 to 95% of people within 5 years of a pandemic have been infected with said virus. So we just assumed with this 2014 cohort, yeah, most of these people have seen it. It should be fine. They have high pre-existing immunity. But in fact, when we went back to the serum from this person, they had no neutralizing antibodies against this H1N1 virus prior to vaccination, and therefore, looked more like the 2009 monovalent vaccine cohort. But just because of the way the study was designed, we didn't move that. And I also want to highlight that this is also true for antibody response against the conserved stop domain. So if you have low pre-existing antibodies against that virus, you are more likely to recall memory B cells against the stock. Okay, so we next wanted to understand, well, there's this process of antigenic drift. Typically, we don't see a lot of broadly neutralizing antibodies within the population. So we wanted to understand, well, is there any evidence of antigenic drift at broadly protective epitopes? And what was really remarkable to us is when we started looking at those RBS and lateral patch specific antibodies, it became clear that while they bound decently well to the 2009 pandemic H1N1 virus, that by 2015 and particularly by 2019, that most of these antibodies no longer bound to these circulating H1N1 viruses, suggesting that perhaps there was antigenic drift taking place at these two epitopes, perhaps because there was such a massive induction of these antibody specificities in response to the 2009 pandemic. So when I was a postdoc, I sort of started to do these in vitro antibody-driven viral antigenic drift experiments, where essentially I could grow virus in a dish in the presence of a monoclonal antibody against a known specificity at a sub-neutralizing level. And I essentially would just keep passing that virus with increasing concentrations of that monoclonal antibody until I managed to generate a virus that that evaded that antibody. So in this case, we were selecting for viruses that were anywhere from about 64 to 128x IC50. So really, really high concentrations of antibody that this virus is evading. And this was a project I started as a postdoc, really was ramping these up in about February of 2020, got most of them made, had to throw them into the freezer in March of 2020. So when I started my lab back in 2022, Marina Good was my first graduate student. She was rotating through my lab. And I said, hey, just go into the freezer, thaw these and sequence them and tell me what's going on. And so she did just that. I think within 3 days, she had these data back to me. So good job, Marina. But anyways, as you can see, there's a variety of different RBS-specific monoclonal antibodies that we had done this with. And there were a few, and just to highlight here in blue, that most of the mutations, again, we're seeing are sort of around the receptor binding site, but not necessarily directly within the receptor binding site. But one thing that really stood out to us were the fact that multiple viruses came back with this S186P mutation, as well as one virus that came back with a D190E mutation. And why that was of interest is that D190 is a direct receptor binding contact. So the receptor for influenza viruses is sialic acid. So there's different flavors of sialic acid. It's a sugar at the end of the day, but we'll keep it simple. It's a sialic acid. And again, D190 directly forms a hydrogen bonding interaction with sialic acid. And then D190 and S186 actually stabilize a water sialic acid contact. So again, another point of stabilization of receptor binding. And what's been really remarkable is if you sort of look through time at these two residues, you can see that around 2019, that a mutation, specifically S186P, was accumulating in circulating H1N1 viruses. And again, by about 2019, this became the dominant, proline at this site became the dominant residue there. In contrast with the D190, sort of shortly after the introduction of that 186 position, you can nicely see that there was a mutation at the D190 position. In this case, it was largely mutating to an alanine. So from a aspartic acid to an alanine is a pretty big change, especially if that side chain of the amino acid as an aspartic acid is hydrogen bonding with sialic acid. You are going to lose that contact if you are now an alanine. You can see it sort of comes up and then eventually goes away. So we wanted to understand, well, are these specific amino acid, these mutations associated with evading RBS-specific antibodies? And so what we did is we just generated recombinant California 2009, so the pandemic H1N1 virus, and we incorporated either this S186P mutation, a D190A mutation, or both in combination. And what we identified in sort of these brackets of different colors here just indicate clonal expansions. So these are from the same clone. And one thing hopefully that you can appreciate is that not every RBS antibody acts the same. So I should also note that we've standardized these data to the sort of Cal09 binding level. So the lighter the color, the more binding you have to that mutant. The darker the color, the less binding you have to that mutant. And so again, you can see here that some antibodies actually increase their binding slightly when there's an S186P mutation, whereas other antibodies really lose that binding as a result of that S186P. In contrast, we have other antibodies that are that lose binding when there's that D190A position, suggesting that this is also an antibody escaping mutation. And then sort of when you put the two together, doesn't really change it too much. So these data to us suggest that, depending on the antibody class or the sort of way that these antibodies interact with the receptor binding site could influence whether S186P or D190A is an antibody escape mutation. And I also want to highlight that there's been some really great work done by Aaron Schmidt's lab, while he was still a postdoc with Stephen Harrison, now runs his own lab at the Reagan Institute in Boston, where he really identified that this sort of class right here, that this like VH459, oftentimes with the V lambda 321, and then sort of have these very specific CDR3, heavy chain CDR3 motifs. This is really the most common way that monoclonal antibodies can interact with the receptor binding site. And again, this class right here really is like that textbook definition of this most common class. And therefore, perhaps this D190A really was accumulated to evade this class, but we can never know sort of definitively if that's true. Okay, so working with our collaborators at Scripps Research, so specifically Monica Fernandez-Quintero in Andrew Ward's lab, we generated a cryo-EM structure of one of these, commonly used RBS-specific antibodies with a Michigan 2015 virus. So this is prior to the introduction of that S186B and that D190A mutation. And just to note, this is sort of just how the CDR3 is making contact with the receptor binding site. But what I've sort of highlighted here on the right-hand side is all of the different contacts of this antibody with the HA molecule, with many of these contacts being on that on this 190 helix. The 190 helix is where D190 exists. And then you can also see right here is that D190 is actually a direct contact of this specific antibody, which sort of makes sense as to why D190A could then evade this, or that mutation could evade this antibody. Okay, so because these antibodies are clearly, because clearly these mutations they're evading antibodies, but they're also affecting receptor binding, we wanted to understand, well, how does this affect receptor binding? And I apologize, my dog is barking at something. I don't know if you can hear it. I don't know what she's barking at. But again, we wanted to understand how are these mutations affecting receptor binding? And so just to note, again, sialic acid is a terminal sugar on a more complex glycan structure. And so what we did is we generated recombinant HAs with either that S186P, D190A, or both. Oh gosh, she's like knocking stuff over. It'll be fine. And we took these HA proteins and we ran them along a glycan microarray. So in this case, we're specifically focusing on Nu5AC, which is one of two types of sialic acids. This is what we have in our bodies. We do not express Nu5GC. We only express Nu5AC, and then tested these. And I also just wanted to highlight, what does a glycan look like? Because prior to really getting into this, I did not know anything about glycans per se. And so again, these sialic acids are, again, these terminal sugars that can either be linked by an alpha-2,6 or an alpha-2,3 linkage. In this case, human influenza viruses use an alpha-2,6 sialic acid. And this sialic acid will be on what we refer to as a glycan core. So oftentimes this is a LACNAC structure. This yellow and blue square would be that LACNAC structure. Oftentimes there's multiple polylacnacs that the sialic acid moiety will then sit on. And then oftentimes while I'm showing something that is sort of balanced, that the arm lengths of the glycan are even, in vivo, certainly these glycan cores can be of various lengths while still having terminal sialic acids on both lengths. So sort of there's a lot of complexity when it comes actually to the receptor of influenza viruses. Okay, so using this glycan array, which includes a variety of different alpha-2,6 Nu5AC linked glycans, we identified that this is roughly the range of the California 2009 HA. When you introduce that S186P, it actually really expands the receptor binding breadth. So this seemed to be a positive aspect of the fact that it could use more glycans with alpha-2,6 sialic acids, potentially as receptors to enter into a cell. In contrast, the D190A, at least in the context of the Cal09 backbone, really reduces the number that you can bind to, suggesting that this may be negative for viral infection. And then what was really surprising, again, on this backbone of Cal09, is that when you put the two together, you actually do maintain this sort of increased binding breadth. But we also wanted to understand this in the context of circulating variants, and this is still ongoing work. If we look at Michigan 15, which has S186 and D190, so not that dissimilar from Cal-09, and really doesn't have any mutations around the RBS, we see that actually there's a slight increase in receptor or different glycans that this HA can bind to. In contrast to the Hawaii 19 virus, this virus has both the S186P and the D190A, so a little bit more akin to this one, but you can see that that binding is really restricted. And so what this really suggested to us is that we need to start putting these mutations or reverting these mutations on relevant backgrounds to really understand how these mutations are affecting receptor binding, and also to confirm that the sort of data that we've generated for the monoclonal antibodies also hold up, and that is D190A on something like Hawaii 2019 necessary, or sorry, that the alanine position at position 190 in Hawaii really necessary for evading this antibody class. And we have that data, and it is true. We're sort of finishing all that up, and I didn't have all the data to show you today, but you know, maybe in the future. Okay, so mechanistically, you know, why is this? What are these mutations doing within the hemagglutinin protein? And so, again, working with Monica Fernandez-Quintero, who is a phenomenal molecular dynamics wizard, I would say, she identified that in the absence of a receptor, this S186 position and the D190 position actually are hydrogen bonding with each other. And what's indicated here in gray and then sort of in black are the two most common conformations of HA. And in fact, when you mutate this first to a P186, which is what happened in nature, this mutated first, you can see that this really disrupts the structure of the HA protein and makes that RBS much more flexible. And that is also highlighted here by the B factor. So wild type, you can see this is sort of the RBS right here, not a lot of flexibility. But as you mutate the S186P, you know, much more flexibility within this region, and also is true with the D190A in the absence of S186P. Okay, so what this really concludes to us is that H1N1 is evolving within the receptor binding site. We think that mutations at S186 probably improves fitness of the virus, and that's why it's sort of been maintained in influenza viruses, whereas D190 was a fitness hit and therefore came up and kind of went away. And we're really trying to understand, you know, sort of how this happened, understand these fitness hits, you know, using viruses to study this. But we also are, you know, trying to understand, you know, can RBS antibodies sort of overcome these mutations in order to maintain sort of neutralization breadth? Because as you can imagine, if you have an antibody that is sort of variant resistant, that targets something like the receptor binding site, then that could really be a very effective therapeutic in the context of like an influenza virus infection, or even as a prophylactic. Okay, so that was my ghost of influenza past, so we'll go on to the present. So, and I'll really highlight actually a study that just got published on Monday or Tuesday, I can't remember what day anymore, where we were really fortunate, myself as a postdoc, to be involved in a phase 1 clinical trial of the first in-human universal influenza vaccine sort of candidate. And the idea behind this is that, you know, as, you know, adults, we all have some level of pre-existing immunity against H1N1 viruses. And as I showed before with this sort of low pre-existing immunity, that if you sort of change the head domain of HA, that perhaps, because this is new, something that you have no pre-existing immunity against, maybe you can recall memory B cells against that stalk domain, so this conserved portion of the HA protein, and by doing like sequential vaccination with these, you could further boost more responses against the stalk domain, and therefore sort of shift these immunodominance profiles. And I just want to highlight that there's been numerous studies that have been published on this, not the one that was just published the other day. Just to note that individuals that received an inactivated influenza vaccine, so inactivated flu vaccines are sort of the standard for seasonal flu vaccines, but this was an inactivated vaccine made using one of these chimeric hemagglutinin or chimeric HA proteins. Again, if it's an inactivated chimeric HA protein vaccine, plus an adjuvant, in this case, AS03, which this was done in collaboration with GSK. So this is AS03 is their adjuvant. You can see very nicely that that vaccine induced a nice response against the stalk domain. So indeed, this vaccine works. So we came in and wanted to address, well, does the chimeric HA vaccine actually induce antibodies against broadly neutralizing epitopes of that stalk domain? And just to note that there are two well-conserved epitopes of the stalk domain. One is the central stalk epitope, which I can't actually point out to because I don't think you can see my mouse, but sort of where I have that unknown specificity binding to, that's sort of where the central stalk is. And then there's also an epitope at the way bottom of the HA protein known as the anchor epitope. So in order to study this within serum, we did this as a serum competition assay. So we can essentially take serum, incubate it with an HA, and then take a monoclonal antibody of a known binding specificity and see how well these serum antibodies outcompete that monoclonal antibody. And so we did this with all the different vaccine samples. And what I'm really showing here is a fold change in titers from pre to post vaccination, either at the prime or then the boost, with the fold change at the boost being at the time of the boost and then approximately a month later. And what, again, just to highlight is that those individuals that received that inactivated vaccine plus adjuvant were more likely to generate serum antibody titers against the anchor epitope and the central stalk epitope. And this was also true for the boosting group. In this case, group 1 was the group that received an inactivated plus adjuvant. So again, this really highlights that this approach can work at inducing broadly neutralizing antibodies against the stalk domain. So then we went on to make monoclonal antibodies from both plasmablasts and memory B cells. In this case, just really day 7 post the priming. And just to note, 2/3 of the response was against HA, slightly lower than what we've seen in other vaccine studies, but still majority HA specific. But when we started to dissect the specificity of these antibodies, we only identified a few against, or I think really only one against that anchor epitope, a few against a trimer interface epitope, which is actually in the head domain. And what was really striking is that we had the vast majority of the plasmablasts were against unknown epitopes of the stalk domain and potentially just unknown epitopes of the HA protein. We couldn't quite figure out if they were binding the stalk or the head. In addition, when we looked at memory B cells, so this should say day 28 post chimeric H5.1. So this is after the boost. The majority of the memory B cells that we managed to pull out were specific to the stalk domain. So that was fantastic. And of those, a very large proportion of them were against that central stalk and anchor epitope, although about a third were against sort of unknown epitopes of the stalk domain. So we next wanted to understand, well, are these antibodies neutralizing? So again, about 60% of the antibodies that we generated were neutralizing. This is specifically from the memory B cells. And if we look at, of the ones that are neutralizing, what epitopes are they targeting? The vast majority of them are against the anchor and the central stalk. And a very small proportion of them, despite being a third of the response, only 10% of neutralizing antibodies targeted these other stalk epitopes. And again, this is just sort of another way to distill this. So about 20% of those targeting these other stalk epitopes were neutralizing. So again, very few of them were neutralizing, whereas all the antibodies against the anchor and central stalk were. But neutralization is not the end-all be-all when it comes to these stalk-reactive antibodies. So we also did sort of a surrogate antibody-dependent cellular cytotoxicity reporter assay and tested this. And what we identified was about 60% of antibodies had this ADCC reporter activity. If we sort of broke this out based on whether they were negative or positive, we found that the vast majority of ADCC negative cells, or of ADCC negative, monoclonal antibodies targeted these other epitopes. And then the ones that were positive were largely targeting the central stalk, which is known to have really good ADCC reporter activity, at least within this assay. And then there's been other studies to show that in vivo, that protection is actually dependent on Fc effector mediated functions. So these data really signify that these antibodies targeting these sort of weird unknown epitopes are neither neutralizing or have this reporter function. I did also go on to do a variety of protection assays in vivo. And similarly, of these antibodies that we put in, none of them mediated protection against influenza viruses. So what are these antibodies against? We spent years trying to get structures with them, and to no avail. We never managed to get a structure with any of these antibodies. And so what we've sort of been hypothesizing what could have happened or what that these were specific to. So what I'm highlighting here in blue and then in green is a study from 2016, just highlighting the structures of H1 and in this case H5. So you can see that this is, these HA proteins, they're trimers, they're sort of nicely kind of stuck together. But when you take the head domain from H5 and put that on the stalk domain of H1, what you can sort of see is that HA head turns out about 60 degrees. And what you're really doing is that you're really opening up that trimer structure. And what we think potentially could have happened with these viruses is that because of the sort of opening of the HA structure, that many of these antibodies targeting these sort of unknown epitopes that we couldn't generate structures with were likely targeting these sort of interface epitopes. And that could potentially be why they're not neutralizing against sort of like a native H1N1 virus, and ultimately did not mediate protection because that epitope may just be very difficult to access in a more wild type virus setting. Okay. Oh, and then also I also wanted to highlight, another challenge of this vaccine is that because when you do place, sort of a novel head on top of this protein, that you also, people make a really robust de novo antibody response against that new head domain. So in this case, a really good induction of H5 specific antibodies, which again, the head domain of this is going to be quite immunodominant compared to that stalk. And as a result sort of a lot of non-neutralizing antibodies in response to this vaccine, it was ultimately, I didn't write this, but GSK dumped the vaccine. And, I think that the sort of readouts that were made were maybe not the most accurate. Neutralization may not be the best way of determining if this vaccine was doing what we wanted it to do. And I think actually doing more in vivo studies and studying these FC effector functions maybe actually would have indicated that this is actually a much better vaccine than sort of the standard influenza vaccine. Okay, so just a little bit of a summary of the ghost of influenza present. So I do want to note that we have an arsenal of broadly neutralizing antibodies to combat potentially pandemic influenza viruses that can be recalled in the context of both, novel exposure to the 2009 H1N1 virus, or in the case of this chimeric HA vaccine. And that we really need to identify vaccines that overcome this humoral immunity against the variable epitopes to induce more broadly protective immunity. And I think we have to really be careful when we're thinking about, immunogen design, right? That we are actually targeting the epitopes that we want to target. And it's hard to know definitively, what proportion of the responses were against the anchor and central stalk epitope and these maybe perhaps other epitopes, but certainly at least within the immune repertoire, there is a lot against those two broadly neutralizing antibodies, and perhaps just a little bit of messing with the structure of this protein could induce even more and more stably induce those responses and sort of prevent the responses we don't want. Okay, so then just to sort of finish off on the ghost of influenza yet to come. And this is not to say that it will come. It's not, it's not certain that it will, unlike death, I guess, which is what the Grim Reapers intended to pose. But it is quite alarming. And why I state that is that we have a major outbreak of H5N1 almost worldwide that is ongoing. This is caused by a clade of H5N1 known as 2344B. This is a high pathogenicity avian influenza virus. So when you hear like HPAI, that's what that means. I also want to highlight that this does not mean that it's high pathogenicity in humans per se. It's high pathogenicity in chickens. That's really what that term means. So if it gets into poultry, particularly chickens, it is almost 100% fatal. So really not a virus that you want to mess around with. Within the United States, this started in early 2022 was when it was first reported. It has been detected in 6 different continents. Remarkably, the one continent left is Australia, and not Antarctica. It was identified in Antarctica last year. All 50 US states have reported cases. This has largely been in wild birds. Over 50 mammalian species have been infected, and then ultimately numerous human infections. And I really need to update this slide. Last report is in the last year. There have been 70 human infections in the United States. And of those, one known death that happened about 2 months ago. And these infections, again, they can range from anywhere from asymptomatic. People don't even know that they're infected. These are largely people that are calling birds to control outbreaks. And then importantly, there's no known human-to-human spread, which is a major barrier for this virus to become a pandemic. But I also want to highlight that because this thing is just sort of burning through the United States, that this can have major impacts on agriculture, which also means impacts on our food supply chain. So this is actually a picture from 2013, not this year. If you guys remember 2 years ago, egg prices were also, at the time, sort of astronomically high for the time. I think that this is actually at Aldi, which if you shop at Aldi, I think prior to COVID particularly, eggs there were maybe $1, right? So $5 for a dozen eggs is sort of really high. And then, now there's, reports, or at sort of the peak of this outbreak, reports of, a dozen eggs being $8, $9, $12 even for just like, regular eggs, not like nice eggs. And then in addition to that, I don't have it written here, but this outbreak has also jumped into dairy cows. And why that's particularly concerning is because there is cow-to-cow spread, and sort of adaptation of the virus to a mammalian species potentially poses it to become more adapted to humans and maybe increases its pandemic potential. Okay, so let's get into the antibodies. So it should be known that we do have some level of pre-existing immunity against H5N1 viruses, and that this is largely mediated through memory B cells targeting conserved epitopes. And so this is highlighting, the study on the left is just highlighting a study from about 2016, I believe, where it was individuals that were vaccinated with this Vietnam 2004 H5N1 virus. This is quite different than the one that is currently causing the outbreak. But just to note that when people receive this vaccine, particularly with AS03, so a nice adjuvant, that they can recall memory B cells against the stem or stalk domain, stem stalk, same thing. And at day 7, so this is really what memory B cell is being recalled. And if you look a month later, you see de novo responses being generated against that head domain. So implying that there's been a new germinal center response against the head. So we wanted to address in my lab if there's any evidence of pre-existing immunity against a 2344B clade of H5N1. And really the question that we asked ourselves, well, can we fish out these rare antigen-specific B cells to understand whether or not we have pre-existing immunity against this novel influenza virus? So we did this with actually just one person because it was the first person we tried and it worked, and revealed some very interesting things. So we just took a 59-year-old woman that donated blood in Denver, and we just studied her steady state PBMCs. And how we did that is that we isolated her PBMCs, we froze them down. Oops. Oh no. Okay, I'm not gonna be able to show the whole thing because it has animation. Shoot. Well, so just to note that, maybe I'll go back to the last slide. So what we can do is that we can incubate PBMCs with an HA tetramer. Each one of the HA tetramers that we use has their own unique DNA barcode on them. So we can actually add a lot of different HA proteins, and then sort of computationally on the back end after sequencing, we can dissect what B cell was binding to which HA. So for this, we purified those B cells that were binding HA. We used the 10x Genomics platform to do single cell RNA sequencing, combined the gene expression VDJ, as well as doing antigen specificity. In this case, we use the Liber-seq approach. So in this case, we use 35 distinct HAs in this study, including seasonal influenza viruses, zoonotic influenza viruses, including actually an HA from the first reported human infection in the United States, which is this Colorado 2022 H5. We also include that chimeric HA and another HA that can allow us to identify B cells against the stalk domain. And then we just had a negative control antigen. So in this case, KLH. If you're a classical immunologist, this is like the word is blanking from my head, but no human has ever seen KLH unless they've accidentally injected themselves with it working in an immunology lab, which hopefully they have not. We sorted class switch memory B cells, and in total, we isolated about 3,000 memory B cells from this individual, with 72 of those binding to the Colorado H5. So this is sort of the data that we get back from the LibreSeq data. So in the middle, I've highlighted here that Colorado H5. So you can see that there's varying levels of binding to this H5. And you'll also notice the sort of the other antigens that are popping up that these B cells can bind to are other group 1 influenza A viruses. It is not uncommon to have B cells that bind to a lot of group 1 influenza viruses. It is rare that you see cross-reactivity to group 2 influenza viruses. And then we're also highlighting here influenza B. So sort of group 1 specific B cells is a little more common. So we went and sort of plucked out some of those B cells that seemed of interest. We made recombinant monoclonal antibodies from them and then just screened them against every single influenza A subtype, as well as a few influenza B viruses. And just to note that we did identify 2 antibodies that were targeting that central stalk epitope, so not terribly surprising. And then we also identified 2 antibodies that were targeting the anchor epitope. What's really remarkable about this 2807 is that it actually bound to every single influenza subtype that we tested. So all 19 influenza A viruses, and also bound to influenza B. And then also sort of surprisingly was this 1358, which had increased binding breadth relative to the other anchor antibody, which was really sort of surprising to us because anchor antibodies that we've previously published on are really cross-reactive within H1, but not necessarily as well at binding other influenza subtypes. So we went on to just sort of highlight this. So again, known anti-anchor antibody breadth, they largely target these individual HAs, so H1, H2, H5, really good affinity against H1 and sort of poorer affinity against H2 and H5. In this case, though, with 1358, we identified that it bound much better to both H2 and H5. But we also saw binding to H6, H17, H18, as well as H19, which is this newest influenza subtype. In addition to that, we found that it tends to be more potent than other anchor targeting antibodies, and also can neutralize H5N1 just perfectly fine. In addition to that, we use that FC or the ADCC reporter assay, as I mentioned before, as well as an ADCP reporter assay. And we also noticed that this antibody has actually really good FC effector functions. And to note that only when we tested these sort of other anchor antibody targeting antibodies in a previous report, only 3 out of 18 antibodies we tested actually had this activity in the reporter assay, potentially suggesting that there's something unique about this 1358 antibody. So just to highlight that this is the structure of sort of traditional anchor binding antibodies at the top. So you can see that they sort of come in at like an upward angle to that epitope, which is quite interesting. They kind of look like an anchor, hence the term. But what's interesting to us is that the 1358 antibody, in this case, this is just a negative stain 3D reconstruction, that the 1358 in the context of H8 actually comes in sort of more perpendicular to the anchor epitope. And perhaps because of this sort of direction or the way that it's approaching the epitope, that maybe that allows the FC of these antibodies to better interact with FC receptors. Because you can imagine if you're a regular anchor antibody and you're already coming up at an upward angle, being able to sort of flip that FC back around to be able to bind to an FC receptor may be quite challenging. In addition, for anchor antibodies, we had identified previously that they use a really restricted repertoire of genes, so just a few VH3 genes and a few V-kappa3 genes. And we also noticed that there was this motif within the kappa chain CDR3, this NWP motif, that was 100% conserved across all anchor antibodies. In contrast with the 1358 antibody, we find that not only does it use a different heavy chain, but that in the light chain, even though it uses a V-kappa315, it has a somatic hypermutation at that N position within that NWP motif, and instead is encoding for valine. And we actually found that if you revert this valine back to that asparagine, that you actually reduce the binding breadth of this antibody. And sort of conversely, if you mutate an asparagine to a valine, in this case, we did it in the context of this 222-1CO6 antibody, that actually increased binding breadth of this antibody now. So we think that this mutation actually could be a way to engineer these anchor antibodies to sort of have increased binding breadth to diverse influenza A viruses. Okay, and then also just to highlight this 2807 antibody, which was also quite interesting. We did identify that it is targeting the central stalk domain, but just to highlight on the far right-hand side, we're showing the angle of approach of other published central stalk targeting antibodies relative to this 2807 antibody. What I think is quite remarkable about this antibody is that it's sort of coming in at a very peculiar angle. In this context with this structure, there were 3 Fabs binding to HA. This is a top view of the HA structure. You can see it sort of makes it look like a propeller almost. So really sort of a really interesting angle of approach. In addition to that, use some genes that I've never seen before for these central stalk targeting antibodies, particularly a V lambda 861. I probably would not be the first person that studies antibodies to highlight that. I didn't even know that there was a VL 861, so that was pretty sort of remarkable to me. And in fact, just to highlight sort of the CDRL2, which is sort of one of the main contact sites of that light chain, that there are extensive contacts within the fusion peptide. But we also noted that there was extensive interactions with this glycan at N154, both mediated by a lysine in the CDRL2, but also this histidine that was created via somatic hypermutation that is sort of pi stacking with that glycan structure. And in fact, we're doing structure, or we're starting to manipulate this HA protein. If you remove that glycan, you really reduce the affinity of binding. So we think that this glycan is maybe a trick that this antibody is using in order to bind to a lot of different influenza A viruses, because this is a really well conserved glycan across both group 1 and group 2 influenza viruses. In addition, we did identify that this antibody is neutralizing, but it was sort of weakly neutralizing. What we did identify using, again, these FC reporter assays is that it had really nice breadth and potency at mediating these functions. In this case, either ADCC for the FC gamma receptor 3A, or ADCP, so cellular phagocytosis via FC gamma receptor 1 and 2A. Okay, so just some take-home messages for today. So as I mentioned, we have this arsenal of memory B cells to combat influenza or pandemic influenza viruses, both highlighted by the 2009 pandemic. We've identified B cells that are targeting H5N1 and also the chimeric HA vaccine. Vaccines that can overcome humoral immunity against variable epitopes can induce broadly protective immunity. So this is true for, again, both 2009 and the chimeric HA. However, careful thought really needs to be taken as to which epitopes are targeted. What is clear is that post-2009 H1N1 has rapidly evolved to evade broadly neutralizing antibodies against the head domain. Just for the sake of time, I didn't go into the lateral patch, but basically lateral patch antibodies are obsolete. They are not, they do not bind to recent H1N1 viruses anymore. And then we also need to be thoughtful of immunogen design in that we are trying to preferentially induce responses against epitopes of interest and avoid responses against epitopes that do not mediate protection. Okay, so with that, I want to highlight the people in my lab that did this work. So Marina Good was the one that's really been doing the antigenic drift story. And then Lauren, Imick, and then Wei Ji are the ones, Lauren did all of the sort of Libre-seq analysis and just a wizard at doing that in bioinformatics. And then Wei Ji, who is a postdoc in the lab that has really been dissecting those antibody responses. Certainly my former postdoc lab, particularly Henry and Carol, who contributed a lot to the chimeric HA study. Alec Frayne at Mount Sinai, who helped me actually initially with making those virus escape mutants. And then we collaborate a lot with Andrew Ward's lab at Scripps. So just want to highlight Julianne and Monica, who we worked with a lot. And with that, I am glad to take any questions.
Thank you so much, Jenna, for your presentation. So a few questions. If you could go back to the slide where you talked about this on a few different slides, but S186P and D190A.
Yes. Sorry, it's just gonna take a bit.
This is perfect. I don't know if this is specific to your research focus, but like what triggers those types of viral mutations, like at that granular level? If that question makes sense.
Yeah, so what would be driving these mutations? And I think at the end of the day, we'll actually maybe go to like this graph,
Yeah.
right? Because at S186 P mutation, right? It's really hard to say exactly what drove this. It should be noted that this mutation has occurred in pre-2009 H1N1 viruses, not necessarily in humans, but in pigs. And so we think that this is probably more adaptive for receptor binding and had sort of, we're still trying to dissect how it's evading RBS specific antibodies. And it may be actually due to the fact of this increased flexibility. So as you can imagine, if you're an antibody trying to interact with this particular portion of the receptor binding site, I realize you can't see my mouse, so you don't know where I'm pointing. But if you're trying to interact with this and this protein is really, really flexible on the surface, that sort of flexibility of the protein, as well as the fact that these antibodies really only mediate binding through their heavy chain CDR3, or mostly through their heavy chain CDR3, you can imagine it may just sort of, fling them off to a degree. So it's sort of like working multiple ways, that it increases receptor binding and maybe flinging these antibodies off. But perhaps for these other ones that are making interactions specifically with like the D190A, maybe you needed that additional mutation in order to evade those responses. And so we are in the process of sort of generating, so certainly we made the 4CO5, so this one binds to the D190 position, but we are also in the process of making one of these antibodies that lost binding due to the S186P mutation. So in this case, this 1AO3 antibody here, and hopefully that will provide some insight as to why S186P can evade this and whether it's a direct contact.
Got it. And so bouncing off of that, like, how are you able to isolate specific viral strains? Like you mentioned Michigan 15 and Hawaii 19, like, how do you know that they're appropriate comparisons to the other influenza strains? Are you tracking like different regions for potential small flu epidemics or?
Yeah, no, that's a very fair question. So the strains that we have been using are actually vaccine selected strains. So they're intended to be sort of representative of distinct clades that are circulating. I did have like sort of a video initially in this presentation that sort of highlighted the different clades as they sort of come up, and also highlighting those vaccine strains, but I had to take it out because I was concerned about trying to run a video. Yeah,
totally. And then one last question. So on slide 19, you referenced some unknown epitopes. You said that like a lot of the epitopes that were binded to were like unknown. So I was just wondering, is it within the scope of your research to identify what those epitopes are?
Yeah, so yeah, we've spent probably 4 years trying to get a structure of a single one of these, and we're never successful at doing so. We also, because they're neutralizing, we don't have the added benefit of like trying to do like those escape mutations, because you can't. They're not gonna. That antibody is not doing anything. It's not putting any selective pressure on the virus. So that's also sort of been a challenge. We did do sort of like these competition assays to try to understand like, well, how many of them are actually targeting the same epitope, for example. And in fact, most of them were not. We did find a handful that's, sort of clustered together, that they're binding a similar epitope, but the majority of them, there's probably at least 5 different epitopes that were being targeted. And again, I think going back to this idea that the HA structure was sort of just open, and just to go to that slide, like it's much more open than H1 or H5. And so it's possible that there's sort of all these different epitopes along the entire sort of length of the HA protein that could be, that these antibodies could be targeting.
Gotcha. And so it sounds like it took a really long time to just identify that in itself. You said 4 years. So trying to identify each individual one would be perhaps a lifetime. So
I think so. And at a certain point, because they're not protective, they're non-neutralizing, these are ultimately things that we don't want to be targeting. And we know that this structure was just not perhaps ideal, that sort of that gives us all the answers that we need. And, we don't need to try to get to the specific epitopes because, again, they're not good targets.
Got it. All right. Thank you so much. That's very enlightening. If anyone else has any questions, please feel free to drop them in the chat. Otherwise, we might be right at time. So I'll give you all a second. Alrighty. Well, thank you again for this very insightful presentation, Jenna. The on-demand version of this webinar will be available very soon, and a link will be sent by email to everyone who's registered. Feel free to watch this or any of our other on-demand webinars when it's convenient for you on the Antibody Society website, as well as our YouTube channel. And thank you again, and have a great rest of your day.
Thank you.